Have you ever wished you could just hit a pause button on a moment? Well, in the world of science, they've basically figured out how to do that, but for life itself. Today, we're going to dive into the amazing and incredibly precise art of cryopreservation. It's the science of freezing living cells to unlock the discoveries of the future. So think about it. The big challenge for any researcher is that the very thing they're studying, living cells, well, they're always busy. They're growing, dividing, changing. They don't stop. So to really study them, or to save a specific batch for later, scientists need a biological pause button. So how on earth do they pull off this kind of suspended animation? Let's get into it. Okay, so before a single cell is even looked at, the whole process kicks off with some serious, meticulous prep work. And this isn't just about being neat and tidy. Every single step we're about to see is designed to create the perfect, sterile, and super-controlled environment. It's all about setting the stage for success. This prep work really boils down to three key things. First, everything gets labeled and pre-chilled. You want everything cold to avoid any temperature shock later on. Second, a special freezing container with the cool name Mr. Frosty gets filled and cooled down. And third, and this is super important, a protective cocktail with a chemical called DMSO gets mixed and chilled. This stuff is basically a lifeline for the cells.
So what is this DMSO stuff? Well, the easiest way to think about it is like a high-tech antifreeze, but for cells. See, when water freezes, it forms these sharp, spiky ice crystals that would just shred a cell's membrane to pieces. It would be a disaster. DMSO is a cryoprotectant. It actually gets inside the cells and stops those deadly crystals from ever forming, keeping all the delicate machinery inside safe and sound.
All right, all of our tools and mixes are prepped and chilled. It's time to get our main characters, the cells themselves. This next part is all about carefully collecting them from the flask where they've been growing, and then concentrating them all down so we can get them ready for the freezing process. And just to give you a sense of the scale here, we're not talking about a handful of cells. In a single procedure like this, a scientist might be handling something like 32 million individual living cells, and every single one of them needs to survive this whole process completely intact. No pressure, right?
So how do you round up 32 million microscopic cells that are just floating around in a liquid? Well, you use a centrifuge. By spinning the tube really, really fast, like 900 times a minute, the force pushes all the heavier cells down to the bottom. They get packed into this dense little clump, which is called a pellet. The leftover liquid gets poured off, and that precious pellet gets put right on ice. We've now arrived at what is, without a doubt, the most important moment in this whole shebang. This is where precision, timing, and technique all have to come together perfectly. Getting this next step right is literally the difference between having a tube full of perfectly preserved cells or a tube of dead ones.
It all starts with some simple but absolutely vital math. See, for cells to survive freezing, they can't be too crowded together, but they can't be too spread out either. There's a perfect Goldilocks density. In this case, it's 4 million cells for every milliliter of liquid. So, since we have 32 million cells in total, a quick bit of division tells the scientists they need a final liquid volume of exactly 8 milliliters.
Now, you don't just dump all 8 milliliters in there at once. Oh no, that would be a catastrophe. It's a really careful two-step process. First, you add half the liquid, so 4 milliliters, of just the normal ice-cold growth medium. This gently breaks up the pellet and gets the cells floating again. Only then do you add the other 4 milliliters, the stuff that has that critical DMSO antifreeze. This slow, gradual introduction is absolutely key, and this is the make-or-break moment. The lab instructions for this step literally have the word critical written in all caps. That DMSO mix has to be added drop by single drop, all while the tube is being gently swirled. If you add it too fast, the sudden chemical change would shock the cells and kill them instantly. So you have to remember, slow and steady saves the cells.
Phew. Okay. The cells are now coated in their protective antifreeze, and they are ready for the final stage, the big chill. But just like adding the DMSO, the freezing part can't be rushed either. We have to lower the temperature in a perfectly controlled way to make absolutely sure those ice crystals don't get any chance to form. And this is where that Mr. Frosty gadget we prepped earlier finally comes into play. Honestly, you can think of it as a reverse microwave. Instead of heating things up fast, its special